Good morning, everybody. Hope you are able to hear me. Okay, just text me so that I can make sure you're hearing me. Okay. Okay. Huda is answering. Yes, we can hear. So, guys, we need to just run, through the topics. Hope we can just get it done quickly. I will just have a certain, run the topic. So, everybody should get ready with your textbooks and some paper. Okay, and just make your notes whenever required, and don't forget to text and ask your doubts if at all you want to explain something in a much more detailed way. Okay. All right. We can just work out. First of all, we'll run and take a revision about all the topics that we have to seriously look into. We'll have to cover both the chapters, so it's very long and lengthy, as you know. But even then, we'll just go fast. So, guys, you come to page number 68, 69, page number, yes, come to page number 70 from there. Only the points we are going to check right now, because otherwise we'll not be able to complete it. Okay. And just inform your friends also that we have just started, by the way. Okay, before others end, if at all you want me to explain some areas specifically, okay, kindly tell that. So you can text it there. You can text us a live chat. According to that, I'll give importance for that area and explain. Remember, we need to cover up everything, including first chapter and second chapter genetics. Only the points. I'll not be explaining in depth, but we'll tell you what are the major things you must cover for your tomorrow's exam. Okay. So, by the way, if at all you have anything to ask, don't forget to ask in the, our text session, message sessions, okay, chat session. Can just chat there, and I will just identify what are the questions you posted there. Accordingly, for that, I'll give importance. And by the way, page number 70, the first point that you have to keep in mind is the Sahiwal cows in Punjab. Okay, so what is that question about? The question is the knowledge about genetics for our forefathers. So Indian forefathers, the scientists had enough knowledge about genetics. For that, we use what? Sahiwal cows in Punjab. So there might be very many other kinds of cows, examples given there, or various other animals given there. But remember, we have to tell Sahiwal cows. Okay. Then Mendel's law of inheritance, under that heading, whenever I'm saying some points, you can circle that points in your book and go ahead, so that you will never miss anything later on, or you can even jot down. Okay, Alina, okay, Arti, Huda, all right. So under Mendel's law of inheritance, Gregor Johann Mendel, what are the basic things he used for creating the law? The answer is statistics and mathematical logic. Okay, that is what the point you need to keep there. Statistics. The mathematics that he used is statistics. Don't use any other area of the mathematics other than statistics. Okay. Then second one, what made his studies reliable? The answer has to be large sampling size. Okay. Remember that if you took experiment in small number of individuals, he's going to get what? Very weak result, isn't it? So if you took only little number of individuals and studied, the result may not be reliable. We cannot trust on such results. So the large sampling size was the most important point we have to keep in mind. See, these particular terms only you will see in MCQ. Okay, what made Mendel study more reliable? Answer should be the large sampling size. Okay, the credibility of the data increased because people could believe it. Then comes what? In that page later on we can come across next paragraph. What is that? Self-pollination, cross-pollination, and true breeding. All this comes at the bottom paragraph in page number 70. So what is true breeding? True breeding means what? Self-pollinated flowers, right? Self-pollinated plants for around minimum 7 generations. So that Mendel made sure that they will make only what particular character. For example, tall plants self-pollinated, so they make tall and dwarf, removed all the dwarf, then take tall, again self-pollinate, they make tall and dwarf, remove all the dwarf, again select the tall. Like that 7th generation he selected the tall and made what? The true breeding. At the end, you remember the true breeding that he made was being test crossed. So don't forget that. Good morning, Atheria. Welcome to you. So true breeding that he made was test crossed, and that test cross to the test cross he made sure what? That the plant is pure for a given character. Next is, with no doubt, guys, you need to remember at any cost, and remember the way, I hope you remember that way I told you to keep in mind the 7 characteristic features of the pea plant, right? So how was that? First, you think the plant as such completely tall and dwarf. That is first point, height, isn't it? So second is what? After thinking about the complete plant, next is thinking about the plant's step-by-step growth. First will be what? When the plant matures, flowers, right? So flowers, where is a flower? Flower axial or terminal. Then the color of the flower, violet or white. So 2 points for flowers. Then once flower become matured, inside what the flower becomes pods, right? So there are 2 points for the pods, which are the inflated and constricted. And then that's a pod shape. Pod color is what? Green and yellow. And then comes what? The seeds inside it. You remember the order? Height, flower, then flower changes to pod, and open the pod you see seeds. So what are the seeds? Seed shape, round ring. Seed color, yellow and green. Look, pod color and seed color, dominant and recessive, are different. So be careful. Okay, pod color, what is dominant? Green, yellow is recessive. Whereas seed color, what is dominant? Yellow is dominant, green is recessive. This might confuse you, so keep an eye on that, circle that. Now, next thing that we have to go about is the first cross that he did, that is monohybrid cross, to reduce the monohybrid ratio, isn't it? So in that monohybrid cross, what all major things you require? Do you remember? I hope you remember it. Even then, what we have to do, I just run around with a single cross and remind you all the major steps that you must have to keep in mind while crossing. So when you write a crossing, it's a must that you have to write what on the top? Monohybrid cross. Hope you are able to recollect this. So if it is a dihybrid cross, you are supposed to write dihybrid cross. And as I told you, right, so when we write this cross, we have to keep what? 2 things, left side margin, right side margin. So on the left side margin, we write phenotype. Okay. For the time being, I'll write everything, but remember, you don't have to write all these things for the exam. Everything is what? MCQ, but you must know what are the order. Phenotype, genotype, then gamete. Here you see gamete, but when we come to F2, it is gamete. Here gamete, because there is only one set of gamete we can get. But in the F2, we'll get 2 kinds of gamete. That's why gametes, the plural is used. Okay. Then F1, then the heading was selfing, remember. And there comes what? phenotype, genotype, gamete, but here is gamete, right? Because plural. Then F2, phenotypic ratio, genotypic ratio. These are the major steps. So, you don't have to basically remember the complete steps right now. Why? Because it won't be asked for exam, but you should have the concept, then only you can clear it according to the what requirement in the exam. So let us go ahead. I think the genotypic ratio, what I have written is not visible there. Anyway, now phenotype, which are they? Tall versus dwarf. Don't use the word short. Kindly use the word dwarf. Genotype, capital T, small t, small t. Gamete, capital T here, small t here. And F1, capital T, small t. And they all were tall. Okay. So we are just running with the revision. So whatever you are catching, the remaining you can sit and explanatory learning you do, but in-depth understanding is required for some points. And whatever you want to like in-depth, kindly let me know through the chat box. All right. Now after that we have to go for what? The tall plant was hiding the dwarf character in it. So to check whether this hidden character is inside the tall plant of the F1, he used it for selfing, right? So phenotype is what? The tall plant is same tall, and here also same dwarf. This plant also is tall. Father and mother both are tall. Genotype is going to be what? Capital T, small t. As it is selfing, both will have the same. Gamete is going to be two kinds: capital T, small t, plus capital T, small t. And then F1, F2 we write with this combination, right? Capital T, small t, capital T, small t, capital T, small t, and small t, small t. So phenotypic ratio is going to be 3:1, and genotypic ratio is what? Genotypic ratio is supposed to be 1:2:1. By the way, guys, what is the most important thing? The questions that could be given from this side to trouble you. We are not using a Punnett square here. So whether we have to use a Punnett square in monohybrid cross, not a must. But if we use, there is nothing wrong. Imagine you are using a Punnett square. How will you write it? So in a Punnett square, you must have what? How many number of products you are going to get? That many offsprings, right? And here we write the male symbol, female symbol, and then we get what? Capital T, small t, capital T, small t. Remember, all the gametes are to be kept in circle so that we can work out very quickly. So Punnett square gives the result like this. Always write capital T first, then only small t. Okay, welcome Shravani, Kiran, and welcome Naima. All right, thank you. So now this is how we make what? Our Punnett square. From there we can collect the details and find out this is pure, this is impure, and pure. So now some terminologies that is a must to understand. That these terminologies you have to seriously focus on. Why? Because based on that, so many MCQs are there. So let me come through all the terminologies he used while doing this particular cross. Phenotype means how a particular organism express its character. The character which has been expressed is called phenotype, isn't it? Okay, guys. Then comes genotype. Genotype is a genetic constitution. See, whatever the character expressed, tall plant may not be purely tall. It might be having that feature of becoming dwarf also, that particular factor. So genotype is a genetic constitution behind the character. A gamete is of course a female or male gamete, that is sperm and egg. And F1 is filial one. The word we used is filial one or generation one or first filial. So why we should remember this term perfectly? Because bracket will have filial, instead of filial there will be what genotype like genial. So many other kinds of explanations will be given there. So you must know the correct name: filial 1 and filial 2. Filial 1 is the first generation. Remember, even though parents are the first generation, we are not giving any letter for them. That might confuse you. Some people might think parent is the first generation. No. So they are filial 1, filial 2, filial 3. Wrong. Parents, we are not giving any name. Filial 1 comes for the first baby set and filial 2 for the second baby set. So then comes what? Phenotype, genotype, gamete, F2, and ratio. Phenotypic ratio, genotypic ratio. Of course, you know both the ratios. And coming to the other terminologies used while he did this experiment, what all things were in his brain. You remember that? Number one, the plant. Suppose the male plant is tall and female is dwarf. He was thinking like if male can decide the character to make the next generation, then I might go wrong. So you remember that point. In two kinds of crosses he did. What is that? Keep that seriously in mind. That term is very important. What is that? The male plant that he suppose he uses a male plant as what the tall. Then there is a confusion. If the male, suppose the male is deciding the character for the next generation, then all my children will become tall because the father is tall, isn't it? So to avoid that confusion, you remember he gave what a particular terminology there. And what was the terminology? That is called reciprocal cross. Very important term, guys. Reciprocal cross. Reciprocal in mathematics is what? 1 by 2 is 2 by 1. Same way, what is reciprocal cross? Catch it sincerely. Reciprocal cross means he did the cross in two ways. One, tall is male, short or dwarf is female. Two, tall is first case tall is male. The second case tall was female and dwarf is male. So he made two sets of what? Crosses. That means tall in one cross is used as male and tall in the next cross is used as female. So that is called reciprocal cross. Okay. So hope that is very clear for you, the reciprocal cross. Now let us go for what? The next point. What is it? While crossing, he used many other terminologies. He was not clear about what makes these characteristics. That time there was no, remember, there was no microscopic, microscopes were not developed or not being in progress. So he doesn't know about the cell, the content of the cell, the genes, nothing. But rather, he used the term what? Factors. That word is a very important word again. So this all I am discussing on view of what? MCQs. So factors. What are factors? Factors are the genes that nowadays we call. So what is a factor? The correct definition is the genes. Today's genes are called as factors. Suppose today's genes are called as factors. That selection is not there in the, in the choices. What is a factor? Factor is the letter he used to represent for something which gives a character from one generation to another. I repeat, the letter he used to represent for something which can give a character from a parent to the offspring. So if he used alphabets, letters, these letters are called factors. Today we call it as genes. So you must remember that term, factors and genes. Then guys, what is next? The next important thing that he used while doing the cross is about, it is about, factors, genes. Then the factors are always in pairs, right? That is what. So the pairs he named as alleles. So people always confuse this. So there is a very simple trick, guys. You look here. There are two L, no? 2L in alleles. When I write alleles, there are two L's. So remember that 2L. 2L means a pair. So a pair of factors. So what is allele, guys? A pair of factors is called alleles for a given character. So what is allele? Definitions you must have in your brain because that will be the question and you have to select from the bracket what is the correct answer. So a pair of factors for a given particular character is called allele. And then the alleles can be either same or different. See, isn't it? So when both the alleles are same, for example, capital T or small t, and when both the alleles are different, so this way alleles can what exhibit. Capital T is what that is called. There are so many names given for this. That's called pure line. One name is pure line. Okay. Another name is true breeding. And the new technical term. Look. So many terms are there for same capital T. Okay. Or capital T, small t. So these same letters are having so many names. Mendel called as pure line or true breeding. And one more name, the new name that we assigned for this one is what? Homozygous. So Mendel's pure lines are dash. So under that, you have to select what? Homozygous. Okay. Then the Mendel told impure breed. That is what with two letters. That's called the new name only. There is no old name for that, guys. So keep in that mind. So heterozygous. So keep making your notes. Don't miss anything. All the points you keep noting down. Then at the end, when you read it, you'll be like, perfect. Okay, I know everything. I'll be trying to go fast with every point. Let us see. But even then, we'll comprehensively cover it. By the way, homozygous, heterozygous, these are the major terms. Remember, homozygous, pure line, true breeding, all are same. All are same. What is the difference? Homozygous is a newly introduced technical term, whereas pure line and true breeding is all term. Okay, and then heterozygous. Then there is one more particular condition called hemizygous. Okay, but that is out of the syllabus. But if at all such a word you are seeing in the in the MCQs, you will certainly get mark for that. Why? Because hemizygous word is not used in our book. But remember, there is one more condition called hemizygous. So homozygous, heterozygous. Then next is what? Why tallness was only found in the first generation? So he introduced another term for that. What is that? So that term should come under this major terminologies. What is that? Dominant, right? Dominant, and the other one is recessive. So what is the definition for dominant? The character which could be expressed in any generation is called dominant. What is the recessive? The character which is never expressed or simply suppressed in a generation is called recessive. So recessive characters, all the 7 characters that we told before, shall we revise? What are they? Number 1, height of the plant, dominant, tall, recessive, dwarf. Now from the plant come to the flower, flower to pod, pod to seed. See the order, okay? From the plant come to the flower. So flower, so height, the position of the flower, axial, terminal. Okay, then color of the flower, violet, white. So violet is dominant, white is recessive. Then from flower we come to pod. Okay, there are 2 pods. The pod color and pod shape. So what's the pod color? Green and yellow. Green is dominant. Be careful. Before also I told that. Yellow and recessive, yellow. Then pod shape, inflated and constricted. Okay, then inside the pod we can see what seed. So there are 2 points for seed also. What are they? Seed shape, that is seed shape is round and wrinkled. The next one is seed color. What is seed color? Seed color is yellow and green. Remember, seed color yellow is dominant, whereas pod color green is dominant. So these are the 7 characteristic features, dominance and recessive, that he studied. All right. So guys, in between, if any question you want to personally ask, don't forget to text there in the chat box. I'll be addressing that. Okay. Now, so from this, after finishing monohybrid cross, why we call this cross as monohybrid cross? What is the answer? Mono means 1. Hybrid means just mixing, right? So one character got mixed up. So tall and dwarf is 1 character. This always people confuse. Okay. How many characters Mendel studied? 7. What is the character? Height. Height is a character. Which are the 2 contrasting features? They are tall and dwarf. Remember, tall and dwarf is not a character. Tall and dwarf is contrasting features, and height is the character. So these words you must keep in mind. So which are the contrasting features of height? Answer is tall and dwarf. What is tall and dwarf? Is it a character? No, it's a contrasting feature of the given character, which is height. Okay. So once we cover all the major topics under monohybrid cross, the most important point that you have to keep in mind is test cross, or also called back cross. So that is our next concept, guys. So let me rub the board and get ready for what explaining you. Test cross. So give the heading: test cross or back cross. Okay. So when we get to topic test cross, what was basically test cross? Mendel wanted to check whether the taken plan for his experiment is pure tall or impure tall. I mean, today's terminology, homozygous tall or heterozygous tall. Look, what is test cross basically? Test cross. Okay. Don't forget to see other videos which I have posted in my channel, which was related to how these genetic problems can be solved. Tough problems and all are solved there. Pedigree solutions are there. So tricks and tips for using such what solution are given there. So don't forget to see them. Test cross. Look, I'll tell you what is a basic, basic reason behind test cross. I have a tall plant. All right. So is it this tall or this tall? That's our question. This is called pure tall. This is called impure or heterozygous tall. Imagine I took this as my parent and started what the cross, then only I get what? I get perfect, correct answer. Imagine I use this as my parent and started, complete cross becomes wrong. Okay. So guys, that is what you should not go to this, but rather take only this. So how Mendel came to know that this is what my plan? looking at a plant, a tall plant, how can Mendel make sure that it is having genotype capital T, capital T? Nothing is written on the plant. On the leaves, it is not written. I am capital T, isn't it? So how Mendel understood that the capital T plant is what I use for my crossing. For that, he designed a special cross called test cross. So what is the definition for test cross? You guys remember, right? What is test cross? Test cross is a cross designed to find the what? We are supposed to find what? The genotype. Okay, so designed to find the unknown genotype of a non-phenotype. How do we say that? So you kindly note down, cross for finding the unknown genotype of a non-phenotype. Phenotype we know, tall. But what is your genotype? I don't know. So a cross to find what is the gene in you or genotype in you. But I know your phenotype. Such a cross is called what? A test cross. What is a test cross? A cross to identify an unknown genotype of a non-phenotype. So that's what we call as test cross, or also called as back cross. So why do we call test cross as back cross? So this term you must remember, guys. So it can be there in what? Our MCQs. Why do we call it as back cross? Why? In test cross, what we do? Look here. So there are two cases in test cross. Let me explain what case 1 and case 2, and then you will come to know why we call it as back cross. In the case 1, we imagine that the plant we use for crossing is pure tall, and we cross it with whom? Double recessive parent, which we use in our what? Monohybrid cross. So that is the first example. So we take this is the gamete, and we get all 100% result will be tall. Now imagine by mistake the plant which I used is heterozygous. So I have two kinds of gametes there, and when I cross them, I get a tall and a short at the ratio 50-50, right? 1:1 ratio. So based on our result only, we identify. So just take the tall plant, do cross it with whom? The small t recessive plant. Just wait for the result. If all the babies are 100% tall, the plant that you used is what? Case 1, that is homozygous tall. Imagine what the cross came out with the result 50-50, tall and dwarf. The plant that you use for the cross is heterozygous. So this is how we understood whether my plant I used is homozygous or heterozygous. Hope it is clear to you. And then next point is why it's called back cross. Look here. This was this or this. You remember this one in our previous cross? This one is the first generation baby. So the first generation F1 is crossed with whom? Mother or father, whatever it is, used in the previous generation. So when the first generation is crossed with the parent generation, from child back to parent, you got the point. That's why we call it as back cross. Okay, so the term back cross comes because the child is crossed back to parent. All right, hope it is clear to you. And if any doubt in this area, you can kindly text me and ask. I hope this area is perfect and clear. If you find everything is okay, or you want any change, or you want any topics to skip and go ahead, kindly text me. According to whatever you say here, I'll be talking. Okay. Otherwise, I'll be going with what? All the points. Now, the next thing is, what are the 3 laws he made? Everybody knows the name of the law: law of dominance, law of segregation, law of independent assortment. But then the 2 laws comes from monohybrid cross. The last law of independent assortment, he could make only from dihybrid cross. Okay, guys. So remember, monohybrid cross has given 2 laws. People always confuse with the second law. Law of dominance, not out for anyone. Okay. Law of segregation, cross spelling is double S. Okay, I'm sorry for that. So law of dominance is always what? Law of dominance from monohybrid cross. Law of segregation also from monohybrid cross. By the way, what is these laws? If you are not familiar with that, let me make it very clear to you. So these laws, you have to have a clear-cut picture. Then only you are going to what? Clearly answer this one. Let's go for law of dominance. It's very easy for everybody. Law of dominance states that what? States that in a pair of characters, in a pair of contrasting features, okay, the one who is dominant will be expressed and the rest will be suppressed. So it's very simple definition only. There is no big technical deeds to understand there behind it. And let's go for law of segregation. That is very important. So what is law of segregate means what? Separate. So guys, law of segregation, you can see over here. Look, when we do the cross itself, you can see the law there. Dekho, here, capital T, small t was the parent. When the parent produces its gametes, he has to separate what the gamete meant for capital T and another gamete for small t, isn't it? So when we do the cross itself, you get what? The law of segregation. So what is law of segregation? At the time of gamete formation, the factors separate or segregate. You got my point? It's very simple. So what is law of segregation? At the time of gamete formation, the factors responsible for a character, whether it is here or here also it segregates, separates. But as they are same number letter, that's why we didn't write two capital T's in circle. It's a rule wise, we have to write both capital T, but we are not writing it because it's same only. No, it will be increasing the number in the ratio. So to avoid it. But why I took this example? Because they are different letters, isn't it? So what is law of segregation? Law of segregation states at the time of, so you have to start from where? At the time of gamete formation, the factors that are responsible for each pairs or the alleles. This pair is called allele now. The factors in an allele undergoes separation or segregation. Okay, and that's all about what law of segregation. Hope that is clear. If you want more explanation, kindly text me. Otherwise, I'll be continuing like that. Okay. Now let's go for what the next one. And for that, what shall we do? We should understand the dihybrid cross. Okay, guys. So it's a very long cross. So I'll be doing it so quickly. And you have to just get the concept. Once the concept is clear, any kinds of MCQs you can easily crack. Okay. So dihybrid cross. So let me write what the abbreviation only. Dihybrid cross. Again, we need to write phenotype, genotype, gamete, F1, right? So phenotype. In dihybrid cross, we use two characteristics, right? Two features. Which are they? You all know it. What are they? One is round seed and yellow seed. Round, yellow, and here it was wrinkled green, isn't it? And what is second one? Round yellow. Round itself is 2R. Alleles rule you have to follow. Some people write 1R and 1G. Finished, gone. Not like that. Round itself is 2R. Every character comes from an allele because everybody is made from a father and a mother. So every character should be in a pair. So round. Yellow also should come as a pair. Okay. Wrinkled, you should not use the letters for this one. Rather use the small letters of rounds. Contrasting is wrinkled. Green is what? Yellow's contrast. So you use small letter YY. Okay. So this is what? Homozygous. This is also homozygous. So gamete. So when 4 letters are there, gamete will have 2 letters. When 2 letters were there, gamete had 1. 4 are there, gamete have 2. 8 are there, gamete will have 4. So gamete will be always having what? Gamete will be having half of the letters for the factors of the parents. So here small r, small y. Then we come across capital R, small r, capital Y, small y. And that's called round yellow. Okay. Now later on we have to go across with what? Selfing. You remember why he does the selfing? Because he knows that some characters are hidden there. To bring them back only, he does selfing. Okay, let us go selfing. What is it? How does it go? Round yellow is capital R, small r, capital Y, small y. Again, capital R, small r, capital Y, small y. So here, so many people commit mistakes. So that could be a question directly in MCQs. Be careful. What is that? Capital R goes with, see the arrows which I draw. The gametes, okay? Phenotype, genotype. Phenotype is what? Round yellow. I didn't write that round yellow here. Genotype is this one. So gamete, capital R goes with capital Y, then capital R goes with small y. Capital R, capital Y is one set. Capital R, small y is another set. Third one, see small r with capital Y, isn't it? And then fourth is small r with small y. So this 4 gametes without any mistake you have to do. Same way, let's go ahead. Capital R, capital Y. Okay, we can do that. Capital R, small y, then small r with capital Y, and small r, small y. So as we cannot do the complete cross with the simply lines, so we need to find this by using a checkerboard. So what is that? That is called the next generation, that is phenotype, genotype, or gamete F1, right? F1. So for that we draw the checkerboard with 16 boxes. Hope you all remember this, right? Just revision, that's why I'm just running through. We will not explain more on that. Draw male here, draw female here. Then write all the gametes, capital R, capital Y, capital R, small y, small r, capital Y, small r, small y, capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. The circle is put to make you sure that the gametes are what? They are the gametes. Okay, so not to confuse them. Now let's go capital R, right? Same pair, capital Y, capital Y, capital R, capital R, capital Y, small y, capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y, capital R, capital Y, small y. Always capital letter should come first. Capital R, small y, then capital R, small r, capital Y, small y, capital R, small r, small y, small y. Capital R, small r, capital Y, capital R, small r, capital Y, small y, small r, capital Y, small r, capital Y, small y, capital R, small r, capital Y, small y, capital R, small y, then small r, capital Y, small y, small r, small y. So after completing this, look over here. You can see all are heterozygous, right? Capital R, small y, small y in this order. Look here, all are what? One character pure, right? Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y, right? So you can see some particular pattern there. Then comes what? Phenotypic ratio. What is that? 9 round. Capital R anywhere you see that is round. Round 1, 2, 3, right? Round yellow. So round yellow, how many? So we'll write round yellow. We get how many? 9. Then round green, we get intermediate in between. No, this is what we was trying to do before. Intermediate character of what? Mixing a character happened. Father was, see, there was no round green in our first generation, but we get round green. Then wrinkle yellow, that is also a mixed character, right? The wrinkle yellow is not here. And then wrinkle green, isn't it? 1. So that is the ratio that we could deduce at the end. But then now from the monohybrid cross, generally, in the dihybrid cross, generally genotypic ratio is never asked. Okay, imagine you got a question. You can just work out this and suddenly write it. But never, ever seen in CBSE's paper till today. So there is no difficulty about that. Now, guys, when you go for what? Monohybrid cross, one question can be found. Many times I have seen that, and that is, what is the mathematical, mathematical reasoning behind monohybrid cross? How Mendel came to this particular ratio? So give your next heading, guys. Mathematical reasoning behind monohybrid, not dihybrid, which I skipped while I was talking there. So comprehensively we have to do. So mathematical reasoning behind monohybrid cross. Okay, write the heading. Okay, let's go for the mathematical reasoning behind monohybrid cross. So what did he do in that monohybrid cross? At the end, he got 3:1 ratio, which was actually 1:2:1 ratio. So this comes by the mathematical identity a plus b the whole square equal to a square plus t square plus 2 capital T small t plus t square. This identity is what he worked out. Okay, so be careful. So a plus b the whole square, a square plus 2ab plus b square. So how we got it? T square means capital T is capital T. 2t was capital T small t, then capital T small t. You remember this was the result he got. Small t square is small t. So the identity used behind is a plus b the whole square. Okay, so that also you must keep in mind. In book it is written as p plus q the whole square. Okay, so everything is same only. There is nothing big deal about the letters or alphabets used. So that's the mathematical reasoning behind it. So why his data was credible? Remember at the starting we told that the credibility of data increased because his example was large sampling size. Okay, now after finishing law of dominance, law of segregation, law of independent assortment, we need to go for a third law. What is that? Law of independent assortment. Okay, So that comes from whom? The dihybrid cross. As I told you, monohybrid cross deduce two laws, which are the law of dominance and law of segregation. Law of independent assortment comes from dihybrid cross. So take page number 80. Guys, come on, go fast to page number 80. And under that you can see the fourth paragraph. What is that? The law states that when two pairs of traits are combined in a hybrid, segregation of one pair of characters dependent on the other pair of character. So many people read it till today, but we are just wondering what does it mean basically. Okay, so what is it? Very simple, very simple. Let me make it as simplified as I can. So in the dihybrid cross, what was the top parents we use? Round, yellow, and wrinkle green. You remember this? Wrinkle green. So these are the pairs of characters we use in a dihybrid cross. So round is one character, yellow is yet another character, wrinkled is the recessive character of round, green is the recessive character of yellow. So we have two sets of characters, right? That's why we call it as dihybrid cross. So now read the definition once again. Law states that when two pairs of traits are combined in a hybrid. That part you got it? So when two pairs of traits are combined means in a dihybrid cross only. That's what that part is saying. Okay, that means this cannot be, or this particular law cannot be taken in a monohybrid cross, or cannot be deduced in a monohybrid cross. So in a dihybrid cross, what happens when two characters are combined? Did we get round yellow into green only? No. Our result was 9:3:3:1, right? So what was 9? Round yellow. What was this one? Wrinkled green. What was this? Round green. This is wrinkled yellow. This is what we have to look into. Okay. So when round yellow and wrinkled green, when two different characteristics were brought together to a hybrid, what happened? While this hybrid was made, this character did not depend on the other character. Round was not at all adamant like I will go only with yellow. Wrinkle was not at all adamant that I will have to only stay with green. No. Round had his freedom to choose either I go with yellow or I go with green. He could even choose green. Wrinkle had his freedom to go with either with what? Yellow or with green. Okay. So segregation of one character is, see the other part, segregation of one character is independent of the other pair of characters. Now you understand what I mean? So round had the independent law of independent assortment. Dependence means freedom, isn't it? Round has the freedom to choose yellow or green. Wrinkle had the freedom to choose what? Yellow or green. Or opposite way you can say. Yellow had the freedom to choose round or wrinkled. Green had the freedom to choose round or wrinkled. All these things are together having the same sense. So what is law of independent assortment? Segregation of inward. This happens only in dihybrid cross. When two contrasting characters are brought together into a hybrid, segregation of one character is independent over the other. They can choose whatever partner they like. How this area is clear to you. Okay. Now let us go for what? Let's go for the next part, that is our what we call, that we skipped in between now, incomplete dominance and codominance. Everything very easy areas only, so there is not a big deal in that. So incomplete dominance and codominance, right? The heading, guys: codominance, incomplete dominance. Okay, so these are simple areas. So let's complete fast. Incomplete dominance. So remember the name of the plant used here: Antirrhinum majus, right? Or Mirabilis jalapa. There are two plants. Okay, in your book we have what? Example of Antirrhinum majus or dog flower. You have to remember the common name also. Why? Sometimes you may get what? Snapdragon. Okay, dog flower, snapdragon, Antirrhinum. Any of these names can be there. One more flower which I told in the class was what? Mirabilis jalapa, right? That's also an example for what? Incomplete dominance. Anyway, study what is given in the book. All three things are same: Antirrhinum, dog flower, snapdragon. Okay, and then let us get to what? Incomplete dominance. What is that? Basically, in that plant, some flowers are red and some are white. So we use what? These letters: red and white flower when we cross, the resultant flower became pink, means blending of character happened. And this goes entirely opposite to what Mendel told. I repeat, what was Mendel's finding? Characters never blend. Okay, characters never blend, they never mix up. But in nature, we saw mixing of character, right? This is very important. Okay, that's called continuous variation. What is that particular term used, guys? Keep that note on that particular term. What Mendel told is discontinuous variation. Be careful these two terms. What is discontinuous variation? Discontinuous variation means characters never blend. One will stand separate from the other. That is what Mendel told. But in nature, what we can see, this is according to Mendelism. But this is what in nature. In nature, we can see continuous variation. For example, human height, you see, are they having only tall and dwarf human? No, they are having so many variations. That's why continuous variations is visible in what human height. But for example, as a character. So that is what in nature we can see continuous variation. Whereas in Mendel's experiments and all, he taught every characters are having discontinuous variation, either tall or short. There is no intermediate. But this is actually something against that rule, and that's why we call it as incomplete dominance. Why? Was this capital R able to show its dominance? No, 50%. Now, what is the genetic reason behind incomplete dominance? That area you must check. What is it? So, as you already know, molecular basis chapter, these genes, capital R. For example, see, let me give the explanation. Capital R gives 50% of the red coloring pigment. For example, let's take what anthocyanin. Okay, anthocyanin is a red coloring pigment. So capital R gives 50%. One more capital R is there, 50 more percentage. So 2 capital R is there. The plant makes 100% red coloring pigment. That's why it was red in color. Okay, now small r gives 0%. Another small r gives 0%. So when both are small r, 0% of the pigment and the plant didn't have any red color. So that was white in color. Okay, now what is next? Now, what is next point? When capital R, which is 50%, and small r, which is 0% mix, we got total expression of 50. And that 50 could give little red color, half of the part red color, the remaining half was not given. So when when in a white paper, you put some red colored, red colored ink and white colored ink and mix. Okay. Paint. When you mix it, what will you get? You'll get a light pink color, isn't it? So the flower became pink in color. So that's a genetic description behind why this happens. Okay. So for knowing that in details, come to page number 76. Come to page number 76. It's very important area there. At the bottom, you can see what theoretically this is possible by 3 points. Did you see the 3 bullets there? Number 1, the normal or less efficient enzyme. 2, a non-functional enzyme. 3, no enzyme at all. How the production of various characters are decided. Here, see 1, a normal enzyme. See, a pair of factor can produce a normal enzyme. For example, here what we told is the pigment and can give the 100% color. Okay, that is a normal case. But second one is less efficient enzyme. Suppose it is less efficient, it may not be able to completely show its normal behavior. So what will happen? Its efficiency reduces. Okay. So third, what is second one? A non-functional enzyme. He produced an enzyme, but the enzyme couldn't function. So even though genes are working, the result of the gene, which is the production of enzyme, is not functioning. So that particular character will not be expressed. Third one, no enzyme at all. What? This gene has got some variation and could not make a working enzyme at all. So all these levels, what can happen? Characters can be controlled. That's why we can never say like what Mendel characters are showing continuous variation. Never. It can be discontinuous variation too. So that's what in nature. Now I would like to take you to a codominance. Okay, that is also very easy area, guys. Okay, so what is codominance? Codominance means what? Here incomplete dominance, half dominant, half recessive. Okay, both were sharing half. In codominance, what is basically there? It's not half. Rather, in codominance, both of them are dominant. I told an example in the class. You might be remembering that one boy is highly strong and ruling the class. Okay. Meanwhile, a new admission comes. He is also equally powerful and ruling the class. When both of them came, big fight. Finally, what happens? Who will win? They both came together and made a treaty that we both will what? Rule everyone and enjoy the class. So that's what, when both of them are dominant, it's called cooperatively dominant or codominance. So basically the heading is codominance, means cooperative dominance. So it's just to understand cooperative, you cannot write cooperative dominant. So here the example what we told was skin color, right? Okay, skin color. So the skin color is decided by 3 sets of alleles. A, B, B, C. 3 sets of alleles. Okay. And the other one is what? B, B, C. So they all have their own names. That's very important in this area. So carefully listen to me. These particular terms are not directly seen in book, but they come always for exam. So carefully listen to me. Skin color. That this explanation we are giving about skin color. Okay. One more example is there. That is about blood group. Later on we'll talk. So skin color A, B, B, C is completely, we call it as the black. Okay. And this is called albino. The term is very important, guys. The black and albino. As there are 6 letters, the one gamete will have half of it, right? ABC. So gametes will have three letters. And then we can have capital A, small a, capital B, small c, B, capital C, small c. And this is what the intermediate called. Okay, these terms are very important. Mulattoes. The intermediate ones are called mulattoes. Okay, so they are the major what? Intermediate ones. There are so many kinds of what possible F2, I mean generations for them, possible characteristics. But then what are the some of them? I would like to arrange them in order and tell you. Look, capital A, capital A, capital B, capital B, capital C, capital C. What happened here? What is codominance here? All the genes are actually quantitatively given for a particular character. Okay, at the end I'll explain what is codominance. Let us complete this. So this all the genes are active. This all genes are showing its presence. So he is called dark, or sorry, it's called black. Okay, the person is called black. All genes are active and making melanin. Melanin is the pigment, you know, coloring pigment of our skin. So when all genes are active and making, he is completely black in color. Now, second one, just imagine one gene is recessive. So one gene is not showing its production, is not that black. Okay, so we can call it as what, you know, nigger or that particular character he comes. Okay, now let's go for what? 2 genes what? Recessive. You notice 2 letters became C. So he is called dark. Okay, he is coming under the category dark. Fourth one is 3 genes recessive. Okay, it can be any gene. Okay, here in the example I am taking capital A, capital A, capital B are dominant and others are recessive. It can be what? A 3 gene, 1 C, 1 small b, 1 A. Okay, like this. This is also an example for the same. No, they are called mulattoes. Or like what Indian people come under this, and that's what wheatish complexion we say, no, but that's basically mulattoes. Now come for what? 2 genes recessive. Rest all 2 genes dominant, 3 genes, 4 genes recessive. So they are called what? They are called fair. Okay, fair. Now let's go for what? A little more. Capital A, small b, small b, small c, small c. Only 1 gene is what? Dominant. Very less amount of melanin is produced, and they are called white. They are called white people. Okay. Last one is small a, small b, small c. So this is basically a disease condition. Okay, in which the person doesn't make any melanin, and finally his body is completely what? Purely, even his hair, you know, eyelashes, everything is white, and most of them can't, you know, even come out of the house because sunburn will happen suddenly, and they are called what? For example, albino. Okay. So these are the various shades of skin color that we can get from this particular feature. Now, what we understand in this case, capital A gene, capital B, capital B, capital C, all 3 sets of genes were what? Showing their presence, and that is called codominance or cooperatively dominant. When all of them are dominant, it gave that color. Okay, but here all are recessive and it was no color. So hope that is very clear. The next and the best example that we have asked in the wait, one more thing. This particular dominance is also called as, you know, there are 3 names for this one. Let us. Number 1, quantitative inheritance. What do you mean by that? The quantity of genes decides the color, right? The quantity of dominant genes. So another one is called multiple allelism. Allele mean what? One pair. Here how many pairs? So many pairs, right? When so many pairs of genes decide a character, we call it as multiple allelism. Okay, or also called as quantitative. Each quantity decides the inheritance, and the common name that we use is codominance. Guys, keep in mind, codominance, quantitative inheritance, multiple allelism, all three are same term. Okay, done. But then quantitative inheritance you may not be able to see in case of some codominance examples. That is in higher level you'll study, but that's not our question right now. But the most important example for codominance is coming under which one? Blood group. Let's go for that. Okay. So what is the example? Human blood group. Codominance or multiple allelism. So what is multiple allelism? More than one allele for a single character. Character is one. What is that? Blood group. How many alleles? Not one pair, 2, 3. Okay, extra pairs are there. So let us take the example. Capital I gene is what? The one deciding blood group. It can have a variant of IA or IB or its recessive small I. These are the three alleles I mentioned. Okay, which are they? I is the main gene for blood group. It can have IA or IB or small I. So now what are the choices? IA and IA. A person gets, what is it? A group. IB, IA and small I. Small is recessive according to Mendelism. It's what again? A group. IB, capital IB. Both are B. So B group. IB, small I. That is again B group. Now third choice is what? IA, IB. Here only codominance we are seeing. A is also dominant. B is also dominant. So the person is AB group. That's how it becomes quantitative. Okay. Last one is both of them like this, and that is O group, right? So in this case, where do we see codominance? At this particular point, IA, IB only. Okay, all right. Now, so hope it is clear. And based on this, some questions were commonly asked in CBSE's paper before. So such questions can be asked this time also. For example, a wealthy couple were killed in an accident. Okay, what happens? One man comes and claims that I am the son of this particular couple. Okay, they were not having any children documented anywhere, but he claimed now that I am son of this couple. I want to take all the money from. Okay, so he is the claim. So by using blood group analysis, we can easily find in a very simple way. Okay, by studying blood group, how do we find whether the claim is right or not? So the couple, the one who got killed in accident. Okay, let us check that question. What is that? Such questions might be there. So be careful, guys. So what is it? One who got killed in accident is what? Suppose A group. The father in that what killed one is A group. So he can be either IA or IA small I. So I would like to take IA small I because there's more choices we can make from there. No, IA means only one chance is there. And the mother is what? B group. I mean O group. Okay, mother is what? O group. Okay, so this person is A group. Mother is O group. Now the claim came is what? AB group. The claimed person is AB group, or the claimed person is what? B group. Suppose these are the things. They cannot make what? See, the only possible babies that they can have or blood groups they can have is IA small I A group and small I O group. They can never make what? AB or B group children. So based on that, we can conclude what his claim is wrong. Okay, they will never have a chance of such what. So such kind of question I have seen as a part of forensic studies, blah blah blah, such questions. Okay, so now after codominance, when we go to that page number 80, 81 and all, people have so much of confusions there, right? Come to page number 80, 81. Genotypes, phenotype. That 80 is correct. 81. What is 81? That is, okay. In 81, we see linkage and crossing over, isn't it? In page 80, 81, we are entering into linkage and crossing over. Some scientists' name are given there. What are they? De Vries, Correns, and Sharma. Kindly note down page number 81, very important. Okay, so before that 81, Dihybrid, Shermark and all, the first paragraph says what were the mistakes happened with Mendel, why he was not being accepted by the world. In that we can see lack of communication, page number 81, circle those words, lack of communication. And then secondly, he told there is no blending, but blending is there, right? Those points. But a third one is the use of mathematics in biology, which was not accepted by the people there, and no physical proof. He told so many things, but there is no physical proof for that. So that area is generally before it was used as what 5-mark questions and all. But in this condition, it could be if this 4 you have to remember why. Out of this question will be like which among the following is not a reason for Mendel's failure. So you must know this 4 reasons exactly. Then only you can find out a person or a reason which is not exactly coming in this category. Okay, guys. So that's very important. Now let us get forward. Dihybrid, Correns and Shermark, and there they were able to see chromosomes and microscopy developed. The world has grown, and people were able to see, and they studied the movement of chromosome at the time of meiosis, and they found the movement of chromosome at the time of meiosis and what Mendel told through his experiments were almost same. And they concluded that into what, in page number 82, you are seeing column A, column B, which is chromosome, which is genes. That's all not big deal. Okay, don't worry. There is no chance of what creating a question. But only one point I want to tell there in that column 82, page number 82, column A and B. What is that? The second row, you see, independent pair segregate independently of each other. One pair segregate independently of other pair. So one pair segregate independently from the other pair. Independent pair segregate independently for other pair. So when we check like that, we are suppose you just go back to what page number 80 and see that law states that what segregation of one pair of characters independent of the other pair. So one pair of characters segregate from the other pair was law of independent assortment from whom? From Mendel. So that one pair example given, column B heading is what? Chromosome, and column A heading is what? Gene. So independent pair segregates is for gene. One pair segregates from the other is what? For chromosome. Okay. Now let us get to what? The most important area, that's the chromosomal theory of inheritance. So by studying the actions of chromosome at the time of meiosis, they concluded into what? Chromosomal theory of inheritance. This theory says what? Whatever Mendel told, Mendel's factors are nothing but genes which is available in chromosomes, and genes are discrete units which is meant for taking the characters to the next generation. Now, where they studied, Thomas Hunt Morgan studied in a particular organism called Drosophila melanogaster, right? So you must remember his name. What is the organism used? Fruit fly. Drosophila is there in the book, melanogaster. Okay, that's the other name, second part of the name, Drosophila melanogaster, fruit fly. Why they use fruit fly? Again, 5-mark questions, you need to check why select the option which is wrong for fruit fly. So what will you do? If you don't know the complete options, you will be in trouble, right? So why fruit fly was used? Okay, we can see, they can be easily grown in synthetic medium. Come to page number 83. We are revising past page number 83 again. What is that? They can be easily grown in synthetic medium. Means fruit fly can be easily grown. Mendel waited 1 year for the result because pea plant is an annual plant by some sativa. But here what? He used only 2 weeks. Okay, synthetic medium life cycle is only 2 weeks, and within 2 weeks we are getting the result. So guys, see the other one. Large number of progeny are there. At one reproduction, thousands of babies are there, and each baby had so many different characters: yellow eye, white body, miniature wing. So many expressed characters which can easily be studied under them. Okay, then sexes are very easy to distinguish, male and female. You see that male is bigger in size. Be careful. In the picture you can see male is bigger in size, female is smaller in size. And then they have what? Heredity. So many characteristics are there which are inheritable. So all these 5 points together. So I repeat, number 1, can be grown in synthetic medium, short life cycle of 2 weeks, large number of progenies, differentiation of sexes, and too many characters. So I gave them in order, right? Arranged them in an order and taught you. What is that? So number 1, just you got a pair of fruit fly. Identify whether we can see male and female. So that is first point. Keep in mind. Yes, we can identify a male from a female. Number 2, are you able to grow them? Yes, we can grow them synthetically outside. Number 3, will they reproduce very quickly? Yes, they make thousands of babies. Number 4, these babies have any special kind of different characteristics and all? Yes, they have very many kinds of inheritable characteristics. So all these things together packed in what? That particular session, Drosophila melanogaster, why we are using it for his studies. So next area is linkage and crossing over, which contains the figure 5.11. Guys, suppose the picture in page number 84, 5.11 comes. Okay, I want you guys to be careful. Look, if this picture comes, any part of the picture can be kept and a question could be designed. So what should you do? Remember, cross A, cross B. The upper layer is what? Parents. The next one is the generation. And here again, the difference. You notice here the black dots are nearby. The black dots are far. That's the only point. The nearby black dot means what? The genes are nearby. Far means they are far. That's the only point you have to keep in mind. Now let me explain. Okay, how do we notice that? If the genes are nearby, they have the tendency to go as such to the next generation. And the genes are far, they have the tendency to split and go to the next generation. And that's called linkage and recombination. Such a simple, simple thing, but people are always confused and I don't know why. Guys, listen to me. Let's go fast for that. Okay. What is the next topic? Linkage and recombination. Isn't it? So linkage and recombination. In that, what is basically there? If this is the chromosome, and here there is a gene, and here there is a gene. They are far. Yet another chromosome. Here there is a gene, and here there is a gene. So at the time of meiosis, they are going to cross over, and chiasma will be formed. Then the crossing nodules will separate, and the chance for them to separate. Who will separate faster or easier? So the chance for cutting in between, crossing over in between, this is very high, whereas this is very low, isn't it? So when the genes, what is this shaded part is a gene. When the genes are nearby, chance for splitting the genes apart is very less. You got my point? When the genes are far, chance for splitting is very easy. Snow baby. Okay, now what shall we understand from this concept? Look. This is the chromosome, and this is another chromosome. At the time of meiosis, in class 11, we learned what is it? The chromosome will undergo crossing. So what happens? This and this branch will cross. So the cross happens at specific points. There are millions of such points. Okay, and they are called crossing nodules. When they cross, this chromosome will switch both of them will switch their hands. Okay, so this chromatin will switch with this one, and at the end when they come out, okay, this is a switched part, and on the other one, this is the switched part. So for example, this one, suppose we completely color as red, at the end, see, okay, this part is red remaining here, right? And this upper red part is being switched and given to him. This particular process is called crossing over. Okay, at this time, genes exchange each other between homologous chromosomes. So at that exchange only, what happens? Linkage is broken. So what is linkage? The tendency for genes to go as such to the next generation without getting splitted. What is recombination? The tendency to separate the genes and go for the next generation. When reproduction happens, only meiosis occurs, right? So that time, this will be splitted and taken. So to understand linkage and recombination, the best example that we can keep in mind is a small thread. Okay, this is a thread having 3 knots. Knot A, knot B, and knot C. This is a thread, okay? And there are 3 knots on it. Knot A, knot B, knot C. Now imagine by using a pair of scissors, I'm cutting what? The thread. Wherever, see, here only if I cut, the chance for separating knot A and knot C will 100% separate, right? Wherever I cut, 100% will separate knot A and not C will separate. Am I right? But now, if I cut, now suppose this distance between this and this is 10, okay, units, and from here to here is what, 90 units, total 100 units. Okay, suppose. So if I cut in between this 90 unit space, two genes will certainly separate. Who are they? B and C. So this not A, B, C are genes. Okay. So if I cut anywhere here, 90% of time, what is the tendency to separate? So I'm talking about recombination frequency. Recombination frequency. First case, 100%. Second case, if I cut at this area, 90%. Right? Imagine I cut this area in between this area. Suppose I cut in between A and B. That means the recombination frequency for cutting there, the chance of separating A and B is only 10%, right? Why? Because they are very near. They are very near. So that is a basic concept behind what? Linkage and recombination. So the genes are very near, they separate very rarely. Genes are very far, they can easily separate. Okay, so now the more the linkage, the less the recombination. Based on this, some gene mapping questions are out there generally, but rarely found in CBSE. If at all you want me to explain, you can ask me at the end. I will explain that. Okay. Now, guys, what is remaining in this chapter? Sex determination. Hereafter, the chapter is getting into what we are going with the maximum pace. But still remember, there are so many things to understand. Let me sit for 2 minutes. Okay, guys. So, what is this sex determination? In that, what are the major points to keep in mind? Take page number 85. Come on, fast. Okay, don't miss any of these points that I say. Page number 85. Okay, in that, major points. Number 1, Hen King. Okay, Hen King. We saw his name, 1891. He traces specific nucleus structure all through spermatogenesis of few insects. It was observed in 50% of sperm received the structure and other 50 did not receive it. Okay, actually, what was that? X chromosome. Some 50% of sperms got X chromosome, other 50 didn't get X chromosome. As he doesn't know what was that structure, that's the fun here. Okay, he doesn't know what was that structure sperms got at the time of meiosis. Like mathematicians call, he called it, let it be X. Okay, it's a structure X. From there onwards, till today we are calling it as X chromosome. Okay, so X chromosome, X body. So he named it as X body, which became X chromosome. Now, coming to the sex determination, what is the major point that you have to keep in mind? Sex determination is generally by sex chromosomes, right? There are specific chromosomes. Some terminologies you must familiarize here for that. That is autosomes, allosomes, etc. So what are they? First of all, how many chromosomes human have got? 1, 2, 3, 21, 22, 23 sets of chromosomes are there, right? So the first 23 set which I drew here with the blue color comes from father. Now let me write the next one. 1, 2, 3, 21, 22, 23 sets are there from a mother also, isn't it? So this is the pair of chromosomes that we generally receive from father and mother. Now, when you come across this, see this one, these chromosomes, the last set, 23rd set is what called sex chromosome, right? And it can be sex chromosomes and it can be either XX or XY. XY decides a male, XX decides a female. You know the details about that. So what are these chromosomes? The 22 chromosomes above are called autosomes. You remember autosomal disorder, sex-linked disorder. This is called allosome or sex chromosome. Okay, these terms are required. Autosomes and allosomes. Okay, the first 22 are called autosomes. The last set is called allosomes. So in human, the sex determination is XX, XY type. For whom? Human. And even Drosophila also the same way. Sex determination is XX, XY type. You notice human and Drosophila same. That's why Drosophila is a wonderful candidate for genetic studies. Now, coming to insect, other insect, XX and X0 type. Instead of male becoming what? XY, there is one missing in a male, right? That's why there are two kinds of sperms, hen king, pounded, hen king. Okay, so that's X0 type insect. So who is this? This is male, this is female. Okay. Now, this is insect sex determination. But coming to birds, we are in confusion. In both this case, male can produce two kinds of chromosomes. Here, female can produce only X now, but male can produce X and Y. So in this case, what we call this is called male heterogamety. Whereas in bird's case, who is heterogametic? It's female. Okay, female heterogameity. So the confusion happens if we use the same letters. Birds in female, birds only can produce different kinds of chromosomes. Okay, so ZZ and ZW. ZZ is the male. Same letters come for male. ZW for female. This is female. Okay, and that's called female heterogameity. That's only in birds. So be careful about that. Why they use these different letters? Answer is again simple. Why? In order to avoid confusion with XX and XY, because everybody by heart had XY in their brain. To avoid that confusion only, ZZ and ZW is taken. Now, sex distribution human, you got it, 50-50, right? The chance of what male and female babies. Mutation, come to that. So many terminologies are there, guys. Okay, then let us go ahead for what? Page number 87. Come fast to page number 87, mutation. Good. Oh, by the way, in between, you remember one point we missed about what? An example with Mendelian inheritance and pleiotropy, everything. Pleiotropy we missed, right? Yeah. What is pleiotropy? Opposite of. Okay, let me just explain that. That's also required. Very important. Before I go here, what is pleiotropy? Opposite of codominance, right? Okay, so let me explain that. Then only we'll go for what the other part. So what was codominance? Multiple allelism. What is that? IA and IB, the genes. Two genes for one character, right? There is one allele, one allele. So two alleles for one character. What was that? Blood group. Now opposite of this, you say two allele for one character, one allele for two characters. If you say it reciprocal, right? One allele, for example, okay, one allele for what? Not blood group. Let's take, for example, allele A. Okay, AA for two characters. That's called pleiotropy. Okay, opposite of multiple allelism. One allele for two characters is called pleiotropy. So, guys, when there is one example in our page number, which page number we skipped it, by the way, that page number is okay. Come back here. Where I'll tell you. Yeah, that is in page number 78. Okay, second paragraph. Single gene product may produce more than one effect. For example, pleiotropy. In that we can see starch grain size. Capital B, big grain. Small b, small grain. Capital B, small b, intermediate grain. Okay, so what is that? 3 grain size are there. So what are they? I repeat. So capital B, big grain. Small b, small grain. Capital B, small b make intermediate size grain. Okay, so intermediate means what? Intermediate means incomplete dominance. So this is example for incomplete dominance. This same genes decide one more character. What is that? That is what the next point. So that genes also meant for what? The size of the seed, round or wrinkled, right? See this what paragraph? Okay, they produce round. This is round seed. This is wrinkled seed, and this is again round because that follows what? Mendelian pattern. Okay, so this is very important, guys. Don't miss it. It's such an important thing. I don't know how I missed it. So see this one. This is what? Big, small, intermediate, which is example for incomplete dominance. Round, wrinkled, and this one is not intermediate. What shape of the seed? That's again round. That is Mendelian pattern. But then now see, it decides two characters. One is big sized grain. One is seed. So size of the grain and seed's shape, both are decided. So two characters from one gene is called pleiotropy, isn't it? So this is an example which shows incomplete dominance, pleiotropy, as well as Mendelian inheritance. So don't miss that. Okay. Now let us go and conclude for that last session, guys. Okay. Then what is that? coming to Mendelian, mutations. Which are the major mutations? Point mutations. Then, what are the major points under that point mutation? Deletion, addition, okay, chromosomal aberration, frame shift mutation. All these terms you must know. Oh, nothing can be left out like that. Okay, addition, deletion, frame shift mutation, then what we call chromosomal aberration. Let us understand each one. So, what is mutation? Any change happening on a gene is called a mutation, and that's the resultant is the mutated gene, and the one who makes the mutation is called mutagen. Right? Mutagens. X-rays, gamma rays, ultraviolet rays, all are mutagens. For example, mutation happens in our gene by using, nicotine, tobacco. All this can create mutation and thus dangerous disease. By the way, mutation means what? If there are so many, letters A, T, C, G, T, A, G. So suppose these are the letters of a DNA. If this C gets kicked out, okay, is removed, C is removed, and A comes here. Okay, that is called substitution. This is same way how we can see in football, right? One out, one in. One in, the other one not out means one extra will be there. No. So this is called substitution. Okay. Now another one. What is that? One more, A joined here. The other one is not removed. One extra joined here. That's called addition. Okay. Addition. I'm talking about various kinds of mutation. Another one, this A got deleted. This is gone. Okay, that's called deletion. Did you get me? All these kinds of changes are called point mutations. So mutation or change happening at a single base pair is called point mutation. Okay, clear. So point mutations are like that. Now all these point mutations make complete frame shift, because as we learned the second chapter, the genes are read in three letters, right? Three letters. So first three, next three, next three. So complete frame will become shifted by point mutation. Now altogether we call it as frame shift mutation. Why it's called frame shift? Because reading is always done by three letters, A, T, C, G, like that. So when one letter is deleted, it's A, T, C, G, like that it goes. The other letter will be read before. That's why called as frame shift mutation. Then next one is called chromosomal aberration. In chromosomal aberration, what happens? Complete chromosome undergoes change. For example, when we draw like this, okay, this chromosome cut here, and the cut part, the cut part just fits opposite way on the same chromosome. Okay, this cap I remove from here and fit here like that. Okay, so such things which happens in big, bulk D in chromosome is called what? Chromosomal aberration. Inversion. Okay, this is called inversion. See, the chromosome is completely inverted. So they are coming under chromosomal aberrations. So now, when we come to chromosomal aberrations, what are they? It's in chromosomal. This is point level. In chromosomal level, what are they? There are two kinds of aberrations. What are they? Ploidy and somy. Okay, there are two kinds of aberrations. Which are they? Ploidy and somy. These people are always confused. Carefully listen to me. What is ploidy? I told you we have how many sets of chromosomes? Two sets, right? 1, 2, 3, 21, 22, 23. So these two sets, I got one from father, one from mother. So these two sets of chromosomes together make what? Diploid. This is called diploid. Okay. If only one set is there, we call it as haploid, isn't it? In case of plants, sometimes one more set enters. I'm not talking about human, okay, be careful. One more extra set, 3 sets are there. That's called triploid. Got it? Sometimes there will be 4 or many sets. That's called polyploid. So that's called ploidy. One complete set of chromosome enters. Not one chromosome, one full set of chromosomes. Set means one full haploid set. Haploid is half. No, this half complete 23 adds up is called what? That's called polyploidy. Such like that. What full half of the set is called haploid. Then one more, both of the set together is called diploid. 3 of them are called triploid, many are called polyploid. What is SOMI then? 1, 2, 3, 21, 22, 23. 1, 2, 3, 21, 22, 23. So sometimes, full chromosome will not be added. One chromosome alone is added as extra. Here, for example, 21st chromosome is added as extra. Okay, so how the 21st chromosome alone came there, that we must know. Okay, anyway, this is called triploidy. So this is called trisomy. What is it called? Trisomy. Okay, even 3 chromosomes are there available for a single place. It's called trisomy. When 2 are there, only 2 are there, what we call disomy. That's very common one. If only 1 is there, these 2 are missing. Only 21 is there, the other one nothing is there. That's called monosomy. If both 21 chromosomes are missing, 0, 0. Okay, one more condition. If 0, 0, both 21 chromosomes are missing, it's called nullisomy. So, so many somies are there. Which are they? Trisomy, disomy, monosomy, nullisomy, etc. So, somy means what? Only one chromosome is missing. Ploidy means what? Full set of chromosomes is added or deleted. Hope it is clear to you. Okay. So, how trisomy occurs? And you all learn Down syndrome is what? 21st trisomy, isn't it? So, what is the reason behind trisomy? Guys, we very carefully listen. Down syndrome, the patients, most of the Down syndrome patients, basically, they all are from where? Down syndrome patients are basically born for parents who reproduce after 40 years. Some parents, by mistake, after 40 years, they give birth to children. Okay. After 40, 45 years, most of the mechanism of our body slowly becoming disturbed and, breaking. Or the cellular apparatus is not that good for doing all these kind of mechanisms. So in that case, when meiosis occurs and a sperm is made, okay, carefully listen what I want to explain here. Carefully listen to me. Suppose a sperm is made 1, 2, 3, 21, 22, 23, 1, 2, 3, 21, 22, 23. These are the chromosomes in a person's body. While making sperm, this blue set and this blue set should separate. That's called law of segregation, as we all know. But then at the time, what happens now? This set separates, this set goes. While this set separates, one mistake happened. This 21st chromosome didn't separate with the other right set. It went along with the left set. Did you understand what I mean? So why this mistake happened? Because he's aged 1. And that is called testicular non-disjunction. That term is very important if it happens in male. And in female, if this happens, if ovum is made like this, it's called ovarian non-disjunction. Kindly write down this term, guys. It might be there in here, MCQ. Testicular non-disjunction of chromosomes in male. In female, it is called ovarian non-disjunction of chromosome. That may result in what? One sperm containing 2 21st chromosomes. Okay. Now that sperm joins an egg. Egg already have 1 21st chromosome. So that child will have 3 21st chromosomes. The sperm brought 1 extra, isn't it? So that is how trisomy occurs. And that trisomy which is happening in 21st chromosome is called Down syndrome. Hope it is clear to you. Okay. So same way, when this went, you remember when this went on the other side, he missed 1 chromosome, 21st chromosome, isn't it? So that particular sperm will have only how many chromosomes out of 23? 22 only. Got my point? So that causes what? If that 22 joins with another set of female from egg, what happens? 23 from her, 22 from him. 1 is missing. So in that 1 missing case is called monosomy. You got me? 1 chromosome missing. If both parents are having this problem, that's called nullisomy. And if both are normal, both parents are giving exactly normal, like healthy, that's called disomy. Hope it is clear to you guys. Okay. Now, let us go for what? Testicular non-distinction, ovarian non-distinction. This can cause various kinds of diseases. Okay. Then, pedigree analysis. Guys, pedigree analysis in detail. One video is there in the channel. Don't miss it. You need to see it. I'm sure once you watch the channel, it is around 15, 20, 30 minutes video. You can solve any pedigree questions. Okay. So I don't want to take time for that. It's already there. Now you come for what? Hemophilia, sickle cell anemia, and phenylketonuria, And before that, chromosomal disorders. Let me complete the chromosomal disorders. Which are they? The complete set of chromosomes arranged as a picture is called karyotype. Guys, suppose you see this picture. You see this picture for exam, this picture. And the question, what is this picture? Answer is karyotype. 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 Okay, so that is first one. Second one, what happened? Aneuploidy means what? Loss of chromosome. Polyploidy, additional chromosome. One word which I missed here is what? Aneuploidy. Okay. One set lost is called aneuploidy. Now, Down syndrome. What are the major? What is the other name for what this disease? Down syndrome is also called Mongoloid EDSE, Mongolian disease, because the patients look like Mongolian people. Okay. So write the name Mongolian disease or Mongoloid EDSE. Okay, this the first explained by a side is called Langdon Down. That's why the name Down syndrome. Langdon Down. By the way, what are the major features? Features are like what? From top to bottom you go. Face, moon face, flat nose, okay, open mouth, long and furrow tongue. And you put the tongue out, very long and so many furrows. Coming to chest, okay, wide chest, short stature. Hands are like what? So powerful. The finger will be like ginger, same way. Finger. Okay. Have congenital heart diseases will be there already. So all these things together, and they look like what? All Down syndrome patients look same in the world, whether he's in any country, all look same. So that is Down syndrome. Then comes the most important, which is because of the problem happening with X and Y chromosomes. That is Klinefelter and Turner. So many people confuse these names and write them opposite. So I don't want that to happen. Down syndrome, they will not make mistake. But Klinefelter and Turner, people confuse always. By the way, guys, it's so simple to understand that. Use the trick which I give you. Okay. Keep listening. What is that? One is caused by XXY gene. One is by X0. So that means what? This came from what? Father gave X or mother gave, suppose mother gave X. Father's testicular distension happened and 10 chromosome. The sex chromosome is missing. So the child gets XO. This one is what? The mother gave X. Father gave both X and Y. So XXY. So, now when we write the name, look. Klinefelter syndrome, Turner syndrome, Down syndrome. What is meant by syndrome? By the way, syndrome. Syndrome is a group of symptoms. Be careful. It can be there as a question. Kindly note. A group of symptoms is called syndrome. Okay, this is called Turner syndrome. Turner syndrome. Look. Turner's, which is a big name. Big name. Klinefelter. So many letters. So big letters. XXY. Turner has only 2 letters. That is small name, less letters. Big name, more letters. So remember like that. Don't confuse them. Okay. Now, what is a Klinefelter? What is a Turner? Wait. The basic thing that you have to look for is X chromosome is meant for both male as well as female character. Okay. Y chromosome is meant only for what? Male character. Got my point? X chromosome is meant for both male as well as female. Every man has one X in him, X and Y. So half female is there inside a man. The remaining Y only makes what? Him as a man. So X chromosome means what? Female as well as male. Y means male. So now looking that, who has Y here? He has Y. So he is a male. Who has no Y? So he is a female. Basically, they are what? Male. So what is Oof. Yes, man. By mistake, this plug came out. Shit. I was going to conclude it. Okay. Are you able to hear me? The live stream with the Moori Poi Pandara Day. Kaliatiabo. The Turkashtamaki.